at the beginning.
Good morning, everyone, and welcome to combined MGH Brigham, I guess MGB, Global Health Grand Rounds. I'm Joe Radigan. I'm the Associate Chief for the Division of Global Health Equity at Brigham Women's Hospital. And I'm very pleased to introduce Dr. Jessica Manning today, who will be our speaker. Dr. Manning earned her medical degree from Emory University and her Master's of Epidemiology from the Harvard School of Public Health. She was a resident in our Global Health Equity track in internal medicine at Brigham Women's Hospital. She lived and worked in Africa and Southeast Asia as part of her training. And in 2015, she became a fellow in the National Institute for Allergy and Infectious Disease at the NIH. She moved the NIAID's Laboratory of Malaria and Vector Research. She moved to the NIAID's Laboratory for Malaria and Vector Research in 2017, and has served as the scientific attaché at the U.S. Embassy in Phnom Penh. She's currently an assistant clinical investigator in Cambodia, where she leads the NIAID's collaborative field sites and laboratories. And she's going to talk to us today about some of her work there and some of her work with malaria, some of her work with COVID. We'll get to hear a little bit of Jess's incredible trajectory over the past few years. So thanks so much, Jessica, for joining us today.
Well, thank you, Joe, and everyone for the invitation. It is exciting to come back and speak, especially since I graduated from the program in 2014. So let me start by sharing my screen. Okay. All right, is everybody able to see my title slide? Great.
Yes.
So as Joe mentioned, I'm an assistant clinical investigator with NIAID's International Center of Excellence in Research in Cambodia. So normally I am based full-time in Cambodia, but I happen to be stateside for the holidays. And so this worked out really well for me to come back and share my journey with you. So what we are going to talk about today is really cover the gamut of what we've been doing the last several years in Cambodia. And I'm using this Cambodian proverb for the talk of my title, which is active hands, full bellies, which, as you can imagine, is related very much to the agrarian lifestyle of Cambodia, that the more you plant in the fields, the more you work with your hands, that when it comes time to harvest, that you will be able to reap more, have more reward, have a full belly. So over the last 5 years, we've been working hard and we've been able to see some of those rewards. So I'm really excited to share that today. So I thought we would start by introducing Cambodia and how I ended up there, as well as how NIAID ended up there. Moving on to some of our community-based studies in Cambodia that are more focused on dengue and arboviruses. And then we're going to talk about our pathogen metagenomics system that we set up in 2019 as part of a Bill and Melinda Gates funded grand challenge that proved very useful once SARS-CoV-2 hit in early 2020 in Cambodia. And we'll briefly describe some of our work in collaboration with the government in response to the pandemic. So to get started, here in Cambodia, which is in Southeast Asia, it's really an area that is undergoing rapid modernization and urbanization. So the population, the urban population has doubled over the last year, and it's going to continue to double over the coming decade. And the problem is that this is fairly unregulated, unplanned urbanization and congestion. So the drastic rise of the urban poor and the epidemics that we see within those communities is growing, from flooding to just general poor disease control to other issues with food insecurity. And so despite the fact that Cambodia is a tourist destination, a very popular one for reasons here seen as Angkor Wat, Cambodia also is tourist destination because of the history of genocide in the late 1970s and early 80s from the Khmer Rouge. And what happened in this period of time is that about a quarter of the population was killed, particularly folks involved in education were targeted. And so now we're missing an entire generation of teachers, doctors, lawyers that would really be leaders and building up the pedagogy in Cambodia. So as I begin to talk about biomedical research and science, just keep in mind that you cannot get a PhD in any of these disciplines in Cambodia. You have to leave the country and come back. So these parts of Southeast Asia, Cambodia, Laos, Burma, where the educational systems are not as robust, are more similar to experiencing brain drain, like is seen as in parts of Africa. So how did we get started in Cambodia? And Joe may remember this from my work when I was a resident, but multi-drug resistant malaria always starts in the Mekong. So ever since the WHO started the chloroquine campaigns in the 1950s, with each new generation of anti-malarials that have come along, the resistance always develops at the Thai-Cambodian border. And so NIH was interested in studying this and started setting up field sites in 2006, right as artemisinin resistance was being discovered. And so from there, they formed a collaboration with the Ministry of Health. And then in 2013, I came along as a GHE resident, deciding to work also here in the Otor Minche region on clinical trials surrounding multi-drug resistant malaria, and then flipped over to NIAID in 2015, and have since set up new field sites in peri-urban and urban areas, and also created this International Center of Excellence in Research between NIAID and the Ministry of Health. So this is where we are. So part of what I do, which is a little different than when I was a resident, is that I actually do run a lab in addition to our hospital clinical field site. So we have a regular biosafety level 2 laboratory with all sorts of the typical equipment you would expect. We run an insectary since we focus primarily on mosquito-borne diseases like malaria and dengue. We have our field sites where we do various entomology as well as patient-centered work. And then this is our district hospital, which is in a peri-urban area about an hour outside of Phnom Penh. But now we have 3 new urban hospitals that we also work in that are not shown here. So it takes a village, and I'm prefacing all of this so you can understand the work we do. We work closely with the National Center for Parasitology, Entomology, and Malaria Control, with the National Dengue Control Program, and now the Cambodian CDC. We work closely with all of our district referral hospitals, and as well as with the monks who run the pagodas, because the pagodas do serve as our community base. And you can see here, we have some of our community nurses seeing children in follow-up at one of our pagodas, which are a beautiful place to work. And this is my team in Cambodia. Everybody here is based in Cambodia, except for 3 folks who are based here in the US who support our laboratory and administrative processing. And without them, none of this would happen, working across 12 time zones and multiple languages. So with that, I want to move into what we actually do in terms of research. And here we are really focused on the vector pathogen host triad. So as you can imagine, here in the field, in resource scarce settings, we're at an increased risk for vector-borne disease outbreaks as well as other outbreaks. So what we try to do is leverage the fact that we have cutting edge science happening at the NIH. And then in the field, we have an opportunity here in Cambodia for a real world setting in which we can begin to understand and study vector-borne disease outbreaks. But we don't want to just study them, right? We want to be able to mitigate. We want to develop tools that are going to improve the outcomes and the health in these communities in which we live and serve. And so what I'm going to talk to you about today are those tools. And so we're really going to jump around a lot, because what we're going to start with is this. And some of you may know what this is, some of you may not, but these are mosquito salivary glands. And these actually are mosquito salivary glands from the Anopheles mosquito, which is the mosquito that carries malaria. And so this all began because of our multi-drug resistant work in Cambodia, where we were constantly cycling through drug regimens. And why do we care about mosquito saliva? And it may be a new idea to some of you, but mosquito saliva is this virtual cornucopia of pharmacologically active ingredients that are vasodilating, anticoagulating, and for our purposes, they're actually immunomodulating all of these cells in your skin and your draining lymph nodes. And so what that does is when you have saliva present in animal models, you can see here in black that the levels of viremia, the levels of virus that you get in the animal's blood is much higher when it's through the bite of a mosquito, as opposed to when you just needle inoculate a virus into the skin. And if we take it a step further and we think about immunizing with vector saliva, then you actually can see that it protects against disease in these animal models. So here we have animals who have been vaccinated with Culex saliva. Culex carries West Nile virus. And then folks who were not. And then you can see here that when you challenge with West Nile virus, the vaccinated animals had 100% protection compared to survival of the mock vaccinated animals. And we see this, across all, something's wrong here, across all animal vector pathogen dyads. So we see it in mosquitoes, we see it in ticks, we see it in sandflies, and we see it in parasites, viruses, bacteria. And so our thought was, well, we have all of these issues here in Cambodia and elsewhere with these emerging vector-borne diseases. So Zika epidemic in 2015, everybody went at lightning speed, and we still didn't have a vaccine candidate in the field for nearly a year. We never know when Aedes aegypti is going to develop new competence for a new pathogen. And here we have multi-drug resistant malaria, where we're constantly cycling the drug regimens every 2 years. What if you vaccinate against the vector instead of vaccinating against the pathogen? And can you use this maybe to have a universal mosquito-borne disease vaccine, or even just being able to vaccinate against a vector and then being able to just give you a leg up in terms of any pathogens that vector may carry. And so this is basically a quick rendition of how the saliva is causing a shift. And if you remember your Th2 immunity, so an increase in IL-4 and IL-10, and then a dampening of your interferon response, really that virus killing response. And ultimately, we see that saliva brings in neutrophils and other myeloid cells that are going to be ripe for a virus to infect and then go to the draining lymph node, and then the virus disseminates throughout the body. But if we can immunize with this supra-therapeutic dose of mosquito saliva, then we can actually prime some type of CD4, CD8 response that we don't really know the exact mechanisms yet. And then the next time you are bitten by a mosquito with a pathogen, you can have a rapid tissue resident memory cell response that's going to create kind of a hostile microenvironment for that pathogen. So it's going to control it in the skin because you're going to have increased interferon response, activated macrophages, and that hopefully you would be blocking some of the pathogen reaching the draining lymph node. So this was our thought back in 2015 when we were thinking that why not consider vaccinating against the vector instead of the pathogen? And now this is a little outdated because there is a malaria vaccine that's recently been approved, but a few years ago, it seemed like it was farther along on the horizon. So we picked 4 Anopheles salivary peptides using an in silico selection algorithm, put those into a vaccine and did a first in human study. So this was back at the NIH. So when I want to talk about harnessing cutting edge experiments at the NIH and bringing them back into Cambodia, this is what I mean, because there's a whole host of discussion on first in human trials happening in developing countries. And there are people for it and people against it. In Cambodia, we don't have the IRB infrastructure to host a first in human trial. So we did this at the NIH Clinical Center, and we showed that it was safe. So this is the first time we've given vector saliva as an immunization, and we showed that it was safe and it was immunogenic. We made our volunteers who were vaccinated made antibodies, and we saw an increased interferon response to help that proof of concept along, that you're thinking about making a microenvironment that's hostile to the pathogen. However, this is here in Maryland, where nobody is getting bitten day in, day out by these vectors, or they're not getting malaria or dengue. And so the question was, what about the real world? So this is where we zoom back to Cambodia, which is where I've been the last nearly 5 years. And we ask how we tease apart these complex relationships when you have these populations that are getting bitten day in, day out, and suffering from these diseases their entire lives. And so here in Cambodia, we decided to set up a dengue cohort that we called the Pediatric Assessment Group of Dengue and Aedes Saliva. So we set this up in 2018. And we wanted to make it as comprehensive as possible so that we were looking at the vector. We were being able to trap and measure vector abundance. We were surveying habitats, microclimates, looking at weather data. That we were also looking at our host in terms of what was their antibody responses to IED's saliva, what were their T cell responses, what did they actually do on a day-to-day basis to protect themselves, larvicide, nets, insecticide, and then did they have any type of red blood cell genetic changes. Like we know that malaria is protective. We know that sickle cell is protective against malaria. If we look at alpha thalassemias in Southeast Asia, do we see any relationship to the development of dengue disease? And then we also are interested in the pathogens. So doing passive and active surveillance of these IEDs transmitted arboviruses, dengue, chikungunya, and Zika, and then also our metagenomics that I'll explain next. So putting all of this together, we started basically geotagging all of our pagodas where we do our patient follow-up, which is in yellow triangles, all of our patient houses in pink. And then not on here is an additional 800 houses where we were doing larval surveys, looking for mosquito larvae, and being able to use all of that to put it into epidemiological models. And so the objective first was to begin to lay the epidemiological groundwork to understand this relationship between saliva immunity and then the development of dengue infection or symptomatic dengue infection. And so really with the seroprevalence, we were looking to see, okay, what types of antibodies, what levels of antibodies do we see to each? And so we started in 2018 and we enrolled in the rainy season. And then every 6 months, we would take sera samples from these 771 children. And then if they got sick, they came in with fevers to the hospital where we rapid tested them and also did multiplex PCR for Zika, chikungunya, and dengue, and then also used metagenomics as needed to identify additional pathogens. And so what I'm showing is the flow chart from the last 2.5 years. And you can see that we ultimately had some fallout during COVID. But what I want to highlight is that the majority of our cohort was dengue naive. So nearly 600 of the kids had not had dengue when they entered into the cohort. And this is exactly what we wanted to see. We wanted to follow this naive population. And then we needed to have enough disease to model. And so as you can see here, we were able to track both symptomatic and asymptomatic disease because we were using neutralization titers at each of these 6-month time points. So we could see who was seroconverting in between who may not have gotten fevers. So typically this is called inapparent or clinically inapparent, but for ease of use, I'll call it asymptomatic here. So they don't present to the hospital. And because we ended up having the largest dengue outbreak in Cambodian history in 2019, we were able to gather enough events to model here. And so as you can see, multiple serotypes of dengue are circulating in Cambodia. This is some of the neutralization data. And we can see here that dengue 1 and then dengue 2 are the most predominant serotypes. And then if we look at the antibodies to Aedes aegypti saliva, as you would expect, they're higher in the rainy season when there's more mosquitoes biting, and it's lower in the dry season. And we don't see any type of age pattern in these colored lines, 2 to 3, 4 to 5. They're all basically overlapped following the same patterns. But we do see individuals highly correlating together, higher and then in dotted lower. And so taking those, looking at dengue and then looking at saliva, how do we begin to put those together to understand if there's a relationship like we see in animal models, like we were able to observe in our phase 1? How can we begin to look at this in the real world setting of Cambodia? And so we're using a dengue naive population. So that's the most important here because you, depending on how much tropical medicine you've had, you may or may not remember that there are 4 serotypes of dengue, and if you're infected with one serotype, the next time you're infected with a different serotype, you actually are not necessarily protected. You can actually have worse disease due to antibody-dependent enhancement or flavivirus interactions. And so it may not be that you are having symptoms due to the virus itself or due to, let's say, levels of salivary immunity, but just actually due to these flavivirus interactions. And so we wanted a clean slate to look at this dengue naive population. So what I'm going to show you is only in folks who had never had dengue. And then we wanted to look at this in a geospatial model so that we could think about the flight of the Aedes aegypti mosquito. And then we wanted to create traditional epidemiological models like most of you are probably exposed to. And so here, as you can imagine, if you have increased salivary antibodies, so increased biting from mosquitoes, then you're going to have increased dengue risk. So this is not a large mental leap. This makes sense. It's what we would expect. And so here in red, when we grid this out by 100 meters by 100 meters, which is the flight of the Aedes aegypti mosquito, then you can see when we overlay antibody intensity in red, and then we put all the dengue cases in as crosses overlaid, you can see the co-localization of the signals. And if we put this into an incidence rate ratio, we can see that you have a 29% increase of having dengue if you are in an area that has a 1 unit increase in the optical density for the antibody levels. And we see this in a dose-dependent manner. So as your antibody levels go up, the number of dengue cases in that grid cell goes up. And that's after accounting for population density and all the other covariates that we collect when we're doing all of these surveys and collecting demographic data from our patients. So let's take it one step farther. And now we can see that actually the children with the highest salivary antibody levels were 64% more likely to have asymptomatic dengue. And this was actually not what we were expecting to see. So this actually suggests, or really makes us ask the question, is there a protective role for these anti-saliva antibodies in the pathogenesis of dengue infection, so that the higher levels you have actually mean that you're creating a more hostile microenvironment and that you're actually not going to have clinically symptomatic dengue. And so this is where we have ultimately ended up in the real world. And so now moving forward, we're trying to ask questions to better understand this phenomenon. And one question that we can immediately begin asking that has implications for the field is that which proteins in the saliva are actually causing this, these antibodies. And this is important because if we can track that, we can think about rapid diagnostics that give us a level of understanding the exposure to mosquitoes so we can identify hotspots. Because when there is an outbreak, there's not enough vector control resources to cover the whole country. Basically, you pick and choose, and you kind of pick and choose at random based on which hospitals are having more cases, but that doesn't necessarily cover the catchment areas. And so what we have done is we've looked and found those proteins that are dominant within the salivary repertoire, and then we've produced recombinant proteins so that we can go back and we can test different areas of our field site to see who has higher responses versus lower responses. And then we can make sure that it's specific enough. You can see here people responding in Cambodia versus the clinical center, that we can prevent disease by going in at the beginning of the rainy season and doing a risk stratification by looking to see if people are responding to these proteins, have these proteins, and then we can allocate vector control resources more efficiently. And then moving forward, if people have other tools for vector control, we can actually evaluate those, because the problem is, and is that I've lamented with the Bill and Melinda Gates Foundation as well, is that using a disease endpoint to evaluate vector control means that you need tens and tens and tens of thousands of people for your sample size, which is just not feasible. And so we end up with all these underpowered trials for vector control in the field, and we don't really know if they work. But if we use a direct measure of your biting from mosquitoes by measuring the antibody levels in your blood, then we can actually tell if we see a decrease or not once you implement a vector control. So these are the two main goals. So we're working on how do we continue to find new vaccine targets, and then how do we develop biomarkers that allow us to stratify risk in the community. So that was a lot. And now we're going to completely change tacks here to the next part of our journey, which is pathogen metagenomics. So in 2018, I was invited to my first director's retreat with Dr. Fauci. And I gave a talk about Cambodia, and I showed this heat map, which at the time in January 2018 had just come out. And the yellow here is showing where the most likely next disease outbreak is going to be. And you can see, and this is based on population density, and it is based on like flora, fauna, biodiversity. And you can see Cambodia is lit up. can also see that Wuhan, China is lit up, India. And this was an important study at the time because it was adjusting for the fact that there's surveillance bias in developed countries, right? We know that the US and Europe has much more sophisticated surveillance than we do, let's say in Cambodia. But so that means they're going to be more likely to detect an outbreak. However, where is an outbreak most likely going to occur, and how do we actually stratify risk there? So I said, we know that we have all of these vectors. We think we have these other vectors. We know they carry all of these different diseases. How are we going to find it in an area where we have kind of a lack of diagnostics? And so if we think about Cambodia or Myanmar, there's so many pathogens and we just don't have the diagnostics. So there's a wastebasket diagnosis, usually of dengue and malaria for everything. And then some serological tests that get done in the major hospitals. So in the cohort I was just telling you about, the dengue cohort, those kids coming in with fevers, that first year, only 10% of those fevers were due to arboviruses. And we did not know what the rest of the fevers were from. And so we applied to be part of a grand challenge from the Bill and Melinda Gates Foundation, an exploration grand challenge to use metagenomic next generation sequencing that was in a platform developed by the Chan Zuckerberg Biohub called IDSeq, now called CZID, to identify those underappreciated vector-borne diseases and just emerging pathogens in general. So we set this up by around September 2019. And what we did, for those of you who are not familiar with metagenomics, is you have a sick patient. We enrolled anybody 6 months to 65 years of age who had a fever of over 38. And when you're doing metagenomics, you're sequencing everything in that clinical sample, including the person's commensal bacteria. And so we collected sera as part of our pilot. And then we've, since COVID, added nasopharyngeal swabs. And more recently, we've added stool. So really, there's no bodily fluid left uncovered at this moment in time. And so we extract RNA or DNA. Usually we extract RNA. We do our library preps. And then ultimately, we were using an iSeq, which is the smallest of the Illumina sequencers. It only gives you about 6 million reads per run. But this is what Gates had provided to 10 developing countries, and we were one of them. And then we use the IDSeq, now called CZID platform, which is cloud-based, and it precludes the necessity of having a PhD in bioinformatics to get the necessary information that you need. And so this is great in Cambodia, where most of our lab staff have associate's degrees in laboratory medicine. Everything else has been on-the-job training in our lab, and they're able to actually do the taxonomic identification of the clinical samples themselves. And so we started with that cohort that I told you about, our dengue cohort, and we expanded this to more than 1,200 persons, cross-sectional febrile cohort, and from a much larger catchment area. And where we are now, since 2019, is that we've enrolled more than 1,000 patients. We've sequenced about 80% of them, and we have about a 50% pathogen detection rate, because remember, we were initially only doing sera, so we were missing a lot of respiratory infections before we started collecting stool and nasopharyngeal swabs. So here is also where we initially started, in this peri-urban area, where you can see here the red is the urban areas, and it's about an hour from Phnom Penh, which is this big dense red area on the right. We've now expanded into Phnom Penh, but what I'm going to show you today is our pathogen landscape from the first 500 patients that we recently published, was accepted into PNAS a couple weeks ago. So you should be able to see it soon, the full paper. But as you can see here, the pathogen landscape is dominated by vector-borne disease, which is what we expect. Which was what we expected. So we were, we wanted to then kind of dig in and understand the vectors a little bit more. And so what I'm going to show you now is I'm going to talk about each of these in the setting of why it really matters in Cambodia. So Zika, Plasmodium knowlesi, dengue, chikungunya. So why do metagenomics? A lot of people were like, why do you need that? Why don't you do targeted PCR? And, you know, the thought is that you don't know what you're looking for, and the ability to pull data from that can only be inferred from sequence data is really valuable. And so this is the hospital where we worked during the dengue outbreak that I told you, which was the worst in Cambodian history in 2019. And so here, really, we would just want to ask the question of why it matters. And I think pathogen metagenomics matters because it allows us to treat patients, populations, and do prevention. And that's because we do make a difference in individual patient trajectories. So metagenomics, our metagenomics is not clinically validated. So anything that we identify, we do follow up with a clinically validated test. And this is probably the hardest part, is getting those type of diagnostics into Cambodia or sending them out as needed. And so the clinical impact that we've had from individual cases thus far, so here's in our hospital, you can see our sign in Khmer, it says, got a fever, ? And this is where we enroll. But our first one is an 8-year-old who had a fever. And this looks like a slam dunk for dengue, because we see that the rapid test is positive for NS1, the major antigen of dengue. We see that the person was RT-PCR positive for dengue. And then when we did metagenomics, we see that there is a beautiful, fully assembled dengue-1 genome. So no questions. But we also saw this little snippet pop up on our sequencer when we did the initial read-through. And we didn't think very much of it. It's an HIV-1. In this metagenomics analysis, part of her sample mapped to an HIV-1 isolate from Vietnam. But this is pretty low, right? We're using an IC. It doesn't have great coverage. There's only 14%. It's 1x. Is this real? Is it not real? And so we went back and we resequenced it. We dug in a little bit more. And then we saw that to a different Vietnamese isolate, we actually could get another snippet of coverage. And then once we put a comprehensive across the whole entire HIV-1 gene map, we saw that this patient had 60% coverage once we pulled from different Vietnamese isolates. And we happen to know, because we work in the community, that she lived in what we call like the Vietnamese house. So this all started to make sense. And ultimately, we did the clinical test, and she had undiagnosed and untreated HIV-1 that was vertically transmitted from her mother, and that also her 2-year-old sister did, and now she's on ARVs now. So while we are primarily focused on a lot of these arboviral epidemics and understanding population, viral population, we can also see how we can make a difference in patients' individual trajectories. The second case I'll tell you about is a 13-year-old female who'd had fevers, aches, and chills for 8 months. And she lives about an hour from the village, from the main town. And this is not part of our dengue cohort. She was just coming in with a fever. And we do have patient photo permissions for all of these. This was actually taken by a New York Times photographer in our piece that profiled our study about a year ago. But she also came in and was positive for dengue on rapid test. And so they told her she had dengue and she went home. But on metagenomics, she had no dengue hits, but a little bit of her samples were aligning with Plasmodium vivax, which normally gets diagnosed fairly easily in Cambodia, but nobody had thought to look for it, or that they had looked for it and could never find it. And what we ended up finding was that despite having negative RDTs, she had a very low level of Plasmodium vivax that had not been treated, and therefore she just kept relapsing from her liver because she had not received any treatment for blood or liver stages of Plasmodium vivax. So we were able to successfully treat her, and she now feels great. So, next point, population. So how do we use metagenomics to impact at the population level? And this is where I'm going to talk about public health action. And so I already mentioned that we had this massive dengue epidemic in 2019, where these are the, since Cambodia started monitoring dengue cases in 2002 after the Civil War, that they never needed to have a scale greater than 0 to 1,600. Even in the 2007 epidemic, the 2012 epidemic, all of our weekly case counts never exceeded 1,600. And then in 2019, we peaked around nearly 5,000 cases per week. So the hospital infrastructure was completely overloaded. And what we were able to do, because we had our metagenomics platform in place, we were able to sequence a ton of the dengue that was happening in this part of Cambodia. And now that we've contributed more than 120, this slide's slightly out of date, 120 dengue sequences, that's more than half of all dengue sequences available for Cambodia. So just highlights the lack of sequencing information from developing countries in this area, which may be one of the good outcomes from SARS-CoV-2 genomic epidemiology, is that sequencing capacity has increased in many different places. But what we can now see is that the dengue 2 that was part of the epidemic in Cambodia is completely divergent from any dengue 2 that had been previously seen in Cambodia. So when we do the phylogenetics and the transmission analyses, we see that the most common related ancestor is actually prior to 1960. So this is the first time that we've had cosmopolitan genotype dengue 2 in Cambodia, and this has implications for diagnostics and treatment, as you can imagine. And we would not have been able to see that or to be on the lookout for it if we had not had the sequence data to guide us. And so we can also see that in the summer of 2020, since there's never a dull moment in Cambodia, that we started getting folks with fever, which is no big deal, rashes, runny noses, and when we did metagenomics, also analyses on these patients, we started to detect chikungunya, which is not part of the national surveillance program. And ultimately, working with our National Dengue Control Program colleagues who monitor chikungunya, they deployed targeted diagnostics, and more than 6,000 people were infected in 15 provinces. So using metagenomics to look for what you don't know you're looking for turns out to be really valuable in the sense of now being able to diagnose and use vector control to mitigate this chikungunya outbreak we had in 2020. And now also importantly, chikungunya has been added to the national surveillance programs. Similarly with Zika, we detected an active Zika case. And previously, Zika had not been detected in more than a decade, despite the worldwide epidemic in 2015, 2016. This led us to do neutralization assays, not only in our pediatric cohort, but some of our other cohorts. And in our pediatric cohorts, we now see that 7% are positive for dengue. And in our adult cohorts, nearly 50% are positive for dengue. Zika, are positive for Zika. And so now we can say that Zika is very much so circulating in Cambodia. And we've now received some additional funding from Merck to begin to understand and characterize these Zika infections in the context of dengue in Cambodia. And then lastly, again, the value of metagenomics is that we have another patient who was a male forest worker, and malaria is rapidly being eliminated in Cambodia, which is one of the reasons that we now study arboviruses as the most significant driver of disease. But we could see parasites on this slide. However, this person's malaria rapid test was negative. Their malaria PCR was negative because in Cambodia, they only test for falciparum, vivax, malariae, and ovale, and not the fifth, Plasmodium knowlesi, which is a type of simian malaria. So it's particularly in non-human primates in Southeast Asia. But we were able to align some Plasmodium knowlesi using our metagenomics platform. And now Plasmodium knowlesi is part of the national surveillance program because we went back and looked at Plasmodium malariae cases, and they'd actually been misdiagnosed, and those Plasmodium malariae cases were actually Plasmodium knowlesi. So this has implications for malaria elimination policy in Cambodia. In Malaysia, where they've announced malaria elimination, they're now having an explosion of Plasmodium knowlesi. So it's really too important to think about all animal reservoirs of disease. So this is really where we are: resource allocation, vaccine deployment, and changing national surveillance. And that's how we use metagenomics in order to drive health improvement in the community. And I am pleased to announce that we were awarded the Phase 2 Grand Challenge. So we've expanded into this area in Phnom Penh over the last 6 months with a focus on neonatal sepsis, typhoid, as well as our continued vector-borne disease work. But really, we never know what we're going to find. It's a totally unbiased, agnostic approach to pathogen detection. And we've also been able to upgrade from an iSeq to a NextSeq, which is a much bigger sequencer that gives us more capacity to run more patient samples than we could before. And so this dovetails into, I'll quickly go through our COVID work. So when COVID hit in January 2020, in Cambodia, we get direct flights from Wuhan. And so we knew that it was only a matter of time. And sure enough, on January 27th, we got our first case in a 47-year-old Wuhan tourist to Cambodia, which was ironically right after they closed the airports in Wuhan, but this guy made it through. And so we used our Metagenomics platform, but here's this, our little iSeq. And this was important from a global health perspective because most people in the developing world are not using, huge sequencers with lots of capacity. They're using these small sequencers. And so the question is, if you want to contribute to genomic epidemiology, to next strain, to variant detection, you need full genome coverage. And how are we going to get it with these smaller sequencers? And so the Chinese researchers released those draft genomes on January 12th. And we knew that it was going to be difficult to cover the whole 30,000 base pairs of COVID. And so we had ordered several different types of primers to help us for whenever we did get our first case. And so the first PCR protocols were released, and that's what we started using. Institut Pasteur was the official COVID testing site in Cambodia, and then we were doing the sequencing. And we were able to sequence COVID within the first 48 hours of receiving that sample in January. And you can see here on the heat map that it was a slam dunk, that only the man, none of the rest of his family, had SARS-CoV-2. But if you look here, we had really terrible coverage. We're not going to be able to submit this genome or look for variants or SNPs. It was only 30% coverage of the genome. And so that's because most of all those 6 million reads in our sequencer were going towards the N gene. So it was an RNA expression problem. And so what we did, as I mentioned, we had those primers. And so we randomly overlaid all those primers across the SARS-CoV-2 genome and then amplified the genome and then put it through our little iSeq and all the bioinformatics steps. And we were able to go from 33% coverage to 100% coverage at 15x, still using our tiny little sequencer. And I think visually, anybody who doesn't do bioinformatics can still appreciate the huge drastic increase in coverage that then allowed us to look for SNPs and upload into GISAID and GenBank. And so we really, in January 2020, nobody in February, nobody really knew what was going on, where was this thing going. So we were just happy that we could contribute. And what we didn't realize was that as SARS-CoV-2 began to spread, people were using our protocol and protocols.io because they also had small sequencers and they needed to understand how to enrich in order to get full genome coverage. And so I also want to highlight, we talk about reverse innovation all the time, moving as opposed going from global south to global north instead of global north to global south. But this was because of the sequencer limitations in Cambodia. Our Cambodian team, we had to develop a way around it. And this got adopted by labs in other developing countries, but it also got adapted back to San Francisco in early March when SARS-CoV-2 hit hard on the West Coast, because they have high-powered sequencers, but this amplification process allowed them to run way more samples than they normally could. And then this ultimately got replaced by an amplicon-based approach that was developed in the US. So it's funny to think now, but on February 2nd, when we were doing our first little analysis of our one SARS-CoV-2 genome in Cambodia, there was 15 genomes available in NextStrain and GenBank. And that was it. And we were like, oh, we found this new SNP, great. Not even contemplating 2 years later now that we would be looking at, 2 years worth of variants and evolution and ongoing pandemic. And so I gave a talk on February 27th in Thailand, the last scientific conference I attended in person. And that was, at that time, 116 genomes available. We had one from Cambodia, 2 from Thailand. And then it was just crazy to think that a year later, there was more than 500,000 genomes. And now, nearly a year after this, we have millions and millions of SARS-CoV-2 genomes. I thank my amazing collaborators in Cambodia who, and within the region who help. And then my amazing team that I've already introduced before. And then lastly, everybody in the US at NIH and at various universities with whom we collaborate, as well as Bill and Melinda Gates and Chan Zuckerberg for funding. And I should thank the GHE program at the Brigham for helping me get it all started and launching my career in Cambodia. So with that, I'll be happy to take any questions.